Thank you for that very kind introduction, Sarah. It's a great pleasure to kick off day two of the summit. And it's a really great pleasure to also share with you some of the work we've been doing in research, as well as Sarah said, this new endeavor in publications, in medical publication, the Cambridge Elements series. So my first talk today, I'm gonna tell you about our epilepsy research center without walls, and highlight some of the work that's being done. I'll focus primarily on the work that we've been doing to functionally characterize a large number of KCNQ2 variants, as well as some of the work we've been doing with patient-derived iPS cells, which you heard a little bit about yesterday. And then we'll take a break where I can take questions, and then I'll come back and join with Sarah Wekhuizen about this Cambridge Elements project. So let me begin by just telling you about what is the Channelopathy-Associated Epilepsy Research Center Without Walls. The designation of a center without walls is one from the NIH, specifically the National Institute of Neurological Disease and Stroke, which has funded a series of these multi-institutional and multi-investigator research programs designed to basically break down barriers and channel a lot of resources into studying a very important topic. We were very fortunate in 2018 to be awarded this center, and I will share the glory with my multiple investigator partners who are located at 10 different institutions as listed here and listed below. This includes 5 universities and 3 freestanding research hospitals, including the Lurie Children's Hospital here in Chicago and the Children's Hospital of Philadelphia, and 2 companies, including GeneDx and Q-State Biosystems. This is our gallery of investigators, and I have to say, this is really the dream team for me. This is a group of people that works well together, shares ideas freely, and is all very creative and collaborative. So we're really pleased to have this opportunity to work together. Our focus has been to do a number of things, just to sort of, you can read this if you'd like, but the big picture is, given the enormous number of genetic variants discovered in ion channel genes, including KCNQ2, associated with various forms of epilepsy, we need to wrap our hands around what is the functional diversity of these genetic variants, what is their functional consequence, not only at the level of the channel, the molecular defect, but what does that defect do at different levels in the brain? So this is a study that's designed to use different scales. First is a large-scale effort to determine the functional consequences of hundreds of genetic variants in primarily sodium and potassium channels, probably some calcium channels as well. Then we'll move to some lower throughput but higher content systems, such as human neurons derived from iPS cells, or individual mouse models of single mutations. And those are lower throughput, meaning we can only study a few variants or few mutations, but we get a lot more information from them. And then across all the systems, whether they're the high throughput or the lower throughput systems, we want to investigate pharmacological responses of these ion channel variants. And that has translational opportunities for treatment, of course. So this is the workflow in our center. We start with a group of experts who look over the landscape or the universe of discovered genetic variants in a gene and prioritize them for our functional characterization studies. And then based on those functional studies, we select some variants to explore in more complex models, such as iPS cell-derived neurons or individual mouse models. And here are the genes that we've started to focus on. KCNQ2, you'll notice, is at the top of the list. And to date, that's been the gene that we've made the most progress in. The center is created around these multidisciplinary teams of investigators that accomplish the main goals of the project. These are coalesced into scientific projects listed here. The three projects are Project One, which myself and Jen Pan run or direct. And here is where we're really trying to discover the function and pharmacology of as many ion channel variants associated with epilepsy as we physically can in the 5 years of our funding cycle. Project Two, which is led by Evangelist Kiskinis, also at Northwestern, but in partnership with Owen McManus, a scientist at Q State Biosciences, we're using human neuron models to model the pathophysiology of epilepsy. And here we've had a big focus on KCNQ2, which I'll tell you about. And then lastly, Project 3, led by Jennifer Kearney, also at Northwestern, and Tasso Sagunis at University of Connecticut, really focus on generating new mouse models of individual variants and genes. We are supported by a team of cores. The first and one of the more important cores is the Variant Prioritization Core, led by Ed Cooper and Ingo Helbig. They are really responsible for selecting which variants we should study. And then once the information is generated on the function of those variants, they are responsible for getting that information into a database for public accessibility. And we've chosen to target the ClinVar database, and I hope that maybe I can say a little more about that at the end. And then the remaining cores are to generate mutations. That's led by my colleague Carlos Venoy, as well as Jen Pan, who I mentioned before at the Broad Institute. And our administrative core, which includes Ann Berg, to try to do community engagement. Our center has a website. Sorry to throw my face up there. It's on the website for some reason. But this is where you can get some information, in-depth information about what we're studying. And there's additionally some links to some studies by Ann Berg, including the KCNQ2 study, and ongoing list of our publications. So I want to just give you a little bit of a glimpse of some of the science we're doing, starting with Project 2, which is focused entirely on using induced pluripotent stem cells, abbreviated iPSC, for studying human neurons from individuals who have ion channel mutations associated with epilepsy and related neurodevelopmental disorders. This cartoon illustrates the workflow of generating those cells, and that starts with a small sample of blood from which we can isolate a group of circulating blood cells that are then reprogrammed to these pluripotent stem cells, and those pluripotent stem cells can then be coaxed into becoming neurons. Here's a picture on the right of some beautifully stained cortical neurons generated in the laboratory. These are immature neurons, and they are probably really not great models of an adult neurological disease. But with the disorders we're studying, using an immature neuron to try to understand the early developmental consequences of mutations is probably an advantage. So we find this model to be really attractive. Now, sometimes there's variability in iPS cells, and it's always been a debate, what is the ideal comparison cell? And we think that is ideally done by correcting the mutation in that cell line back to the normal sequence using the gene editing tool CRISPR, so that you have now a parallel cell line that has the same genetic background, but the mutation's been corrected. And that's been our approach from the beginning. Once we have that pairing of the patient-derived iPS cells and the neurons we can make from that, paired with the corrected genetically identical cell line, we can then use those cells for a variety of experimental approaches. And they are just shown in cartoon form here. Some of these are electrophysiological by direct recording from neurons one at a time, or there are other approaches, and I'll show you some examples of multi-electrode array recordings, which can interrogate a small population of neurons and their interconnectivity. And then, because of our collaboration with Q State Biosciences, we have access to a space-age technology called the OptoPatch, which performs cellular electrophysiology on thousands of neurons at the same time using optical signals. And I won't show any data from that at the moment in this talk. We've had for several years now a collaborative program with the Lurie Children's Hospital to aggregate blood samples on children primarily and some young adults who have KCNQ2 mutations as well as other gene mutations. In our current registry, we have more than 20 KCNQ2 samples. About half or a third, probably about a third at this point, have been converted to iPS cells, and several of these have been studied at least preliminarily. One of them, this particular one, arginine 581 to glutamine, is one that we've studied in depth, and this is work that's just been published earlier this year, and it was really the project and really the efforts of Dina Simpkin, who's a postdoctoral fellow working jointly between my lab and that of Evangelos Kiskinis, as well as supported by a host of other individuals. This paper actually highlighted several things. First of all, I think it was the first really in-depth investigation of KCNQ2 developmental and epileptic encephalopathy using this cellular technology. From that study, we learned several things. Two important things. We really defined the electrophysiological behavior of neurons with a KCNQ2 mutation. But beyond that, we found that there were some secondary events that probably are important to know about. Importantly, the focus of this paper is highlighting the fact that in addition to dysfunction of the KCNQ2, KCNQ3 channel, there is another channel or family of channels that's also dysregulated in some way, probably as a byproduct of the primary defect in KCNQ2. This is important because this may contribute to the neuronal dysfunction in the disease and may come on later in the disease and might be a modifier of the condition. And so this is sort of the first evidence of a secondary physiological event that has occurred. Some of the data in this paper is illustrated here. This is just an illustration and a taste of microelectrode array recording, which was done in the Kiskinis laboratory. This is a method in which the neurons are directly plated onto an array of tiny electrodes, which can make contact with the surface of the neurons and detect their electrical activity, either as single neurons or usually in populations of neurons. And so this is a good way to interrogate a population of neurons and understand how they're talking to each other. And each blip here illustrated in these what are called raster plots indicates sort of a signal from that cluster of neurons sitting next to an electrode. And what we learned from this was that in the cells that were identical genetically but did not have the mutation, there was a certain basal firing pattern that we could observe. But in the mutation line, we saw a completely different pattern. And there was a tendency for the neurons to all fire in these sort of coordinated burst-like patterns. And that was significant to us, and that was formed the initial characterization. And there's a lot more information in the paper, and I'm sorry I don't have time to go in depth into it, but just to give you a little taste of what is possible to do. We're certainly now moving forward and doing the same kind of analysis on a larger number of variants, and I think probably our goal is to probably study 8 or 10 different variants that can be for this study. Now, even studying 8 to 10 variants in iPS cells is a lot of work, but that number pales in comparison to the entire number of KCNQ2 variants that have been identified. If you just look in the published literature, there are now well over 400 mutations that have been described. Several of them, probably close to 200 and, well, a little more than 280, are simple amino acid substitutions in the gene. And so these are the ones that give us the most trouble to try to predict their functional outcome. If you look in ClinVar, which is a publicly accessible database that warehouses information largely from genetic testing companies, but also research laboratories, there are over almost 1,400 KCNQ2 variants, and almost 550 or so are simple amino acid substitutions. That's a similar number to what's in the RIKI database. Beyond these these first three columns are all, first three rows, I should say, are all disease-associated variants in KCNQ2. But keep in mind, there are a lot of other variants in the gene that are carried by people that don't have any apparent functional consequence. And these are curated very nicely in a database called NOMAD, which is data from some 125,000 individuals for whom exome or genome sequencing has been done for research purposes. And so that's a large number of variants as well. And this often some of these are very rare, found in only one person in these studies. And so these are a little bit hard to know whether these are capable of causing a disease or whether they're really inconsequential. And then I list up here Anne Berg's natural history study, and we're collaborating closely with her to ensure that we study every single unique variant in that study to enable future genotype-phenotype correlations. Now, any variant on this list can be classified by a standardized approach that assigns the level of probability that a variant will cause epilepsy or related neurodevelopmental disorder by these 5 terms. So highly probable means we would call it a pathogenic variant, and this is what sometimes we synonymously call a mutation. A variant for which there's not quite as much support for pathogenicity, we will call likely pathogenic. And it's these first two groups that we're most interested in. Then on the other end of the spectrum are variants that are so common in the population, or they have no functional consequence that we know of, that we call them benign or likely benign. But unfortunately, in the middle here is a large category of variants for which there's just not enough information to tell whether it's pathogenic or not. And these are called variants of unknown or uncertain significance. And that's really where our study, I think, is making the biggest contribution to try to unravel some of these variants. Just so you know, in ClinVar, here's a pie chart illustrating all 1,390 variants that were registered as of yesterday. There's a large number that are pathogenic and likely pathogenic, representing probably totally about 40% of all the variants. But there are many that are characterized as variants of unknown significance. If you just look at the missense variants, and again, these are the simple amino acid substitution variants, the number of variants of unknown significance, or VUS, and those that for which there's really conflicting interpretation, that represents, those two groups represent half of all the variants, or about 240. So this is where this is where the problem lies. And so if we can add value by performing functional studies on these variants could potentially be classified more accurately. So this effort of ours, hopefully, among other things, will benefit the accuracy of genetic testing. So I'll spend the rest of the time talking about our effort to do large-scale functional characterization of KCNQ2 variants using a tool called patch clamp electrophysiology. This is a tool that's been around for many years in neuroscience. It's really the gold standard for recording electrical activity in a cell, including a neuron, or a cell that's been into which has been introduced a genetic variant of an ion channel like KCNQ2. But it's a slow, low throughput method. Even under the best circumstances, where you have a team of really crack investigators, such as part of the people in my lab, working really hard every day, they can maybe crank out 16 measurements a day of high quality data. That's just not enough to tackle hundreds of mutations. So several years ago, we decided to repurpose an instrument that was developed for drug discovery in the pharmaceutical industry that uses an automated approach to patch clamping. It uses a completely different strategy to patch clamp, and therefore it can be done in 384 well dishes or plates. And this is the instrument in my lab. It's officially called the SynchroPatch, but we've nicknamed it the DOS machine. And in fact, it's so sophisticated, it has its own Twitter account. So if you want to amuse yourself, I don't know if it's very amusing, but feel free to follow it. Traditional patch clamp is really good for sampling one cell at a time. This is a cell membrane with some ion channels in it. And this is the apparatus that allows you to interrogate that single cell and get some readouts, such as what's shown below here for a sodium channel. Automated patch clamp uses a completely different strategy where now the cells are what moves instead of the electrode. And therefore, you can do this in 384 arrays. The instrument we have can actually handle 2 plates at a time, so 768 measurements possibly at a time. So think about this as sort of comparing an old reliable locomotive to a bullet train, for example. So for now, I want to share with you the work we've done so far with this approach. Just yesterday, we finished off a manuscript that I hope to be submitting this weekend, if possible, that describes the functional work we've done, both functional and pharmacological work we've done on 81 different KCNQ2 variants. The variants are listed here to the left. They're color-coded based on what the clinical information that we had available to us, whether they were associated with developmental and epileptic encephalopathy, benign familial neonatal epilepsy, the self-limited form, or whether these were just rare population, mostly rare population variants. And this is the distribution in the ion channel protein itself. The way we developed this assay system, we put a lot of thought into this to adopt this high throughput approach. We first started out recognizing that to study KCNQ2 accurately, to model it as if it were in a brain cell, we needed to provide the cell with KCNQ3, its obligatory partner. So we made a cell line in what are called Chinese hamster ovary cells that express KCNQ3 all the time. And then into those cells, we can introduce a plasmid that encodes the KCNQ2 channel. In this case, we would provide it the wild type channel. And then we would reconstitute what would be accepted as the functional unit of the potassium channel, which is a 4-subunit, symmetric 4-subunit structure. In this case, I've just drawn it arbitrarily as having 2 KCNQ2 wild type subunits in red, and then 2 of the KCNQ3 subunits in blue. And this is not the only possible configuration, but this is the one we think is the dominant one. When we do that and introduce that into the automated patch clamp, we get something like this, which is almost every one of these 384 wells has a current recorded that resembles the KCNQ2, KCNQ3 potassium channel. Now, of course, we want to study variants, so we can do the same thing with a genetic variant of KCNQ2, engineer it into this plasmid, transfect it into the stable cells, and then we would reconstitute a variant KCNQ2 plus KCNQ3, which would resemble if somebody, a patient or a person only had the variant. In other words, they were homozygous for the variant. They had 2 copies of it. And that's not common. What is most common is heterozygous. And so to model that, we just simply introduce another copy of the wild type channel. So now we have the variant of KCNQ2, the normal or wild type WT KCNQ2, and KCNQ3 all together in this complex system. And we can reconstitute this heterozygous model. So we moved on to do some validation work. We started out with a set of variants that were carefully selected by Dr. Cooper to represent the variants that have already been studied and published. So these were our benchmark variants that allowed us to assess whether this new technique was accurate. We also then looked at a group of mostly rare population variants that we expected not to have any functional defect. And these would basically allow us to validate the system in our ability to differentiate a benign variant from a disease-causing variant. And then lastly, we tackled a group of nearly 40 variants that have never been studied, or at least weren't published at the time we selected them. And all this work together required that we make recordings from nearly 9,500 individual cells. That is a lot of work, and we're pleased that we can now try to get that published. In addition to these 39, we have another ongoing batch of cells or variants that we're studying that numbers around 50. And this should include all of the remaining variants from Ann Berg's natural history study that we haven't studied to date. Let's show you this validation study. So just to orient you to how the data are presented, I'm going to show two functional properties that we evaluate in all of the variants. One of them is the current density. That's simply how much potassium channel activity did we see, and that's current, electrical current flowing in the cell. We express that as a proportion of the normal or wild type channel. So 100% would be normal. Anything less than that to the left is a loss of function. Anything to the right would be a gain of function. The other property we look at is a little more sophisticated, and that is the midpoint, the voltage at which 50% of channels are activated. And so we measure that, again, relative to the wild type channel and make basically what is the difference between wild type and the mutant. And again, we've organized this data so that if values fall to the left, it implies a loss of function. And if it falls to the right, it implies a gain of function. So for the validation set, here are the data. We studied these variants both in the homozygous condition, which is not directly relevant to a patient, but gives us a first glimpse at how it works. And also the literature data is available in that state. So here we simply compared the data that we have for different variants, either BF&E variants or DEE variants, red and blue respectively, blue and red respectively. And the black bars represent the data that we could take from the literature. And I would say, for the most part, the data align very nicely. There's a couple where we think the data are not concordant, but very few examples of that. The same goes for the heterozygous condition. So we felt very good after this that our assay was giving accurate results, concordant with what's been studied elsewhere. And so we proceeded to study the 39 variants that have never been studied before. These were, again, mostly DEE variants in red, with a few of the self-limited BF&Es. We first studied them as homozygous variants, and what you should really recognize here is most of the variants show activity that is plotted to the left, meaning loss of function, both for current density, less so for activation. Notice also there are some variants that we would characterize, for example, R144W. When we look at current density, we get a significant loss of function. But if we look at the difference in activation value, it's actually in the other direction. It's in the gain of function direction. This was mentioned briefly yesterday, that some channels have this confusing mixture of functional properties. And I think that's something we have to reconcile ultimately in a more complex model, like a neuron or a mouse. And so that's just something to be aware of. This is the same set of variants, but now studied in the heterozygous condition. And again, there's a number of variants that have loss of function, even in this context, some of which have activity levels that are below 50% of normal. And that's a threshold below which we would consider that variant having a property called dominant negative effects, meaning the mutation is sufficiently bad that it poisons the entire channel, even though there are normal or wild type channels present. Lastly, for every variant that we study, the last experiment that we do on those variants is to apply the drug retigabine, which is relatively selective for activating KCNQ2, KCNQ3 potassium channels. And as many of you know, this drug, which is also known as ezogabine in the US, has been marketed by SmithKline and French, GlaxoSmithKline for several years, but then it was withdrawn from the market and is now really difficult to get. Hopefully there'll be some opportunity to bring it back to life, and I think there's a talk later today from Xenon to give us an update on that. But retigabine is very potent and activates, this is the KCNQ2, Q3 channel under normal no drug conditions. And then in the presence of retigabine, you get a much larger potassium current, and that's its job. So we wanted to know what was can retigabine restore normal channel function for some of these really severe loss of function variants? And also, what would retigabine do to a gain of function variant? And then, of course, there were a lot of variants that had mixed functional effects. So our main goal was to compare what does the normal channel look like in the absence of drug, and then compare the heterozygous channels with one variant copy and one normal copy. How does that channel look in the presence of retigabine, and how do these look together? So here I'm going to show you the data a little differently. I'm going to show you the effect of the drug on channel behavior in what's called a current voltage plot. So here is along the horizontal axis of these plots is the voltage at which we tested the activity of the channel. And on the upright or vertical axis is simply the value of the current that we recorded. Now we set everything on a scale so that the wild type channel or the normal channel had a value of 1 at its maximum level. So this is the way the channel looks without any drug in it. This is a particular variant that I'm going to highlight for you, R581Q, which is the same variant we studied extensively in iPS cells. If we look at now the effect of the heterozygous variant in the absence of the drug, this is what we see. And so there's a considerable loss of activity, suggesting again a dominant negative effect on the channel. And then when you add retigabine, you can restore that value in very effective ways. And depending on the voltage where you look, that effect may be very comparable to the normal channel or might even be even higher than the wild type channel. So this is one pattern that we have observed. Here's a couple of other patterns. Here is a mutation that had very severe loss of function and a dominant negative behavior that didn't respond very well to retigabine. Again, depending on which voltage you look at, you might come away with the idea that maybe it's responding pretty well down here at these voltages, which may be physiologically relevant, but up here in these sort of extremely positive, it doesn't look so good. I'm not sure the physiological significance of that. And then, completely opposite to that, here's a variant which has a mild, maybe a very mild gain of function effect, but it's really not that different. But the effect of retigabine is monstrous. It has a huge effect. And what would that do? We don't know. So I just use these to illustrate for you that we've observed that the retigabine response is quite variable among the epilepsy-associated variants. And there really needs to be a way to assess what is the clinical significance of that. So it would be nice to know if individuals with these mutations or with any of these mutations that we've studied and how they respond to retigabine. And so ultimately, you know, this is just half of the data that we generated. We're now wrestling with how to get these data into the public domain. One approach, of course, is to publish the data, which we're going to do. And if possible, I'll also put the paper pre-publication onto a preprint server to make it accessible almost immediately. But we're also exploring and actively working to get the data into the ClinVar database so that we can, if you click on a variant in ClinVar, you'll have access to a summary of the functional consequences of that variant. So just a brief summary before I take questions. My big picture is that, of course, KCNQ2 is a frequent cause of channelopathy-associated epilepsy. Some of the talks yesterday emphasized that. Many of the variants, unfortunately, have unknown clinical significance. We've been using this technique called automated patch clamp to decipher or decrypt these variants of unknown significance and help to perhaps classify some of those variants more precisely. And then lastly, and I think importantly, and more work has to be done on this, many of the variants we studied exhibit this variable response to retigabine, and the significance of that clinically really needs to be sorted out. This is the team of my laboratory and the list of people who contributed mostly to this work. Evangelos Kiskinis has his own lab in our Department of Neurology, and my colleagues at Lurie Children's Hospital, Ed Cooper at Baylor, and Ingo Helbig at Children's Hospital of Philadelphia. Thank you very much. I'd be happy to take a few questions.
Thank you so much, Al. A couple are coming up in the chat group. I'll start with this one. My son has severe ASD, LGSID, but I don't see his variant in this study. How can I learn more about targeted therapies for his missense mutation? And how can we get his data into research in order to help?
Well, it's a great question. There are so many variants to study. We continue to struggle with how to prioritize them. Our center grant really is devoted to variants associated with epilepsy. However, we are interested and certainly try to examine variants that are associated with non-convulsive phenotypes. I think at this point, we would be very willing to accept nominations of variants from families. At a minimum, we can check to see if we're already working on it. It may not be one that I listed, but again, we have 50 more that I didn't list. And we'll be probably making another batch of 50 sometime in the new year. So anyway, someone can email me if they would like, or email.
I was just going to say, this calls for an email. Do you want to pop that in the group chat, or do you want to say it now for everybody if they've got their fingers poised on keys and want to write it down?
You can. Can I do the group chat in? Can I chat by Zoom, since I'm not doing the streaming right now? So
no worries. You know what? I tell you what, we can get. How about this group chat? We will get it from Al and pop it in for you. We're going to keep going to another question, but yes, we will get that. You tell us whatever the best one is that you want us to have people write it to, and we will take care of that. Let me go, if I can, and ask another question. This is from another mom in the group who says, you say this is the gold standard. We learned yesterday that one variant can both be gain and loss of function depending on what measures are used to determine. You did mention this, Al. You talked about the fact that that can be the case. So could you expand, perhaps, Al, a little bit more on the significance and how patch clamps can work and whether or not that's absolute?
Well, yeah. So this, this binary classification of loss versus gain of function is an oversimplification. We measure a lot of properties of ion channels using this technique, and some of them really don't look abnormal. Some of them look like they are loss of function, and others look like they're gain of function. And it's not unusual to find a variant that has mixed properties. That's at the molecular level. What probably now matters is that we translate that into what is the effect on a cell? How does that impact the firing of a neuron? And I think that's one of the goals of our center, is to try to be able to translate those observations about the molecule and its properties to higher order things. So it is complicated. I don't think that's really independent of the technique used to measure the variant. And I will just say that, again, this binary classification of loss and gain has value, but it can oversimplify the situation, and some variants are just more complicated than that.
Let me ask you another question regarding AI. So, you showed us your cool computer that basically has its own social media account. That sounds like a lot of fun. But how significant, how important is artificial intelligence as you're looking, at lots of different variants, lots of things that you have to kind of tease out? How much a part of the equation is AI?
At the moment, it's not. However, it is something we will approach in the future. I think ultimately, once we have enough data, and I think if we had studied, several hundred variants, we can now put into a machine learning algorithm the clinical features of that condition, and their maybe some information about their drug responsiveness, and the individual details about each variant, and maybe we can learn something that way. That we are not recognizing by just looking at the data ourselves. So I think that's going to be something we want to do in the future.
All right, so that'll be very interesting. And again, we want to make the point that we put your email address is there. I've just checked, Al, it's there in the chat room. So if other people who in our community want to make sure that they can have their child's individual case looked at to see if it's been taken care of, that's there. I've got one more question for you before we let you go and move on to Sarah Weckes, and well, we'll have the two of you. This is from India. My son is on medicines clonazepam, rivaroxaban, sorry if I didn't get that right, carbamazepine, pyridoxine. 6 months old now, and still his seizures are not under control, not responding to voice actions, etc. These cases, I think what's coming through to me from that is, there's a huge burden, isn't there, on this, from this disease. So if we look globally at what you're hoping to do with CWOW, with your Center Without Walls, with all this extraordinary research and your great team, what is your goal, Al? What are you hoping to achieve with all of this work and this research that you're doing?
Yeah. Well, it's a great question, and the person who posed the question, again, we're sincerely, sorry that you're struggling with this. It's a big challenge, of course. Our big goal, I think, first of all, I think we want a better understanding of the diversity of functional consequences of genetic variants in KCNQ2, as well as other genes associated with epilepsy. We think that information will advance and improve accuracy of genetic testing and advance our knowledge of the mechanisms of the disease. And through that, hopefully, we can design more precise treatments for this condition, and some treatments that may depend on what variant a person has and how that variant behaves. So ultimately, we would really love to be able to contribute to being able to stratify patients by function and pharmacology, and that would help hopefully identify the best treatment sooner in the course of someone's illness. Because we all believe that I think treating earliest is the best medicine.
Yeah, I'm conscious of time, so I'm going to just quickly ask one more question, and then we want to move on to Sarah Weckusen. The other question is that we're hearing from a parent in the parent group that Ingrid Sheffer spoke yesterday about the fact that a variant can be both gain and loss of function. I know you spent a little bit of time on this already, but just trying to understand that variant, that it can look like both gain and loss of function, depending on what measure you use. How do you tease out which one it's more likely to be? I understand that they can be very complicated and it can be an oversimplification, but for parents who are trying to decide, for example, certain medicines are great for loss of function and really bad for gain of function, to just give but one example. So how do you definitely decide which it is?
Right. Yeah, this is a really great question. It applies to things that we do all the time. So again, you have to separate sort of these terms at the molecular level, where it's easy to see how a channel can have mixed properties. But what does that dysfunctional channel do to the firing of a neuron? So the cellular level. And so I think assessing how that channel impacts the cell is the next big question. We're trying to address that with these iPS cell neuron models or mouse models. But at some point, maybe a computational model of a neuron would be valuable as well. And so that's a strategy that people are using for other genes and perhaps for KCNQ2. So that's another approach. So I think we have to assess the molecular defect and the cellular defect separately.